10 liver-destroying foods you should avoid eating too much of. When we talk about keeping our body fit, we often focus on the heart, muscles, and brain. But there's another organ that deserves just as much attention: the liver. This powerhouse filters out toxins, aids in digestion, and manages nutrients, playing a critical role in keeping us healthy. Some foods, while delicious, may be true villains when it comes to liver health if consumed excessively. Eating too much of these foods may lead to fat buildup, inflammation, and even long-term damage such as cirrhosis or liver failure. By understanding which foods to watch out for, you may protect this vital organ. That's why, in today's video, we will talk about 10 foods that may damage your liver silently. But do you actually understand why liver damage is a serious health issue? Stay tuned until the end of the video to learn more about the impact of liver damage. 1. Alcohol. Excessive alcohol consumption is notorious for its damaging effects on the liver. When alcohol is metabolized in the liver, it produces a toxic enzyme called acetaldehyde. This enzyme may cause cellular damage leading to inflammation and scarring, known as fibrosis. Over time, repeated liver injury from continual alcohol intake may lead to irreversible changes in the liver, resulting in cirrhosis. Cirrhosis significantly hampers the liver's ability to function properly, impeding its ability to filter blood, fight infections, and process nutrients, hormones, and drugs. Another harmful effect of excessive alcohol on the liver is the development of fatty liver disease, or hepatic steatosis. This condition occurs when fat builds up in the liver cells, largely because alcohol metabolism disrupts the normal breakdown of fats in the liver. Fatty liver associated with alcohol consumption is the earliest stage of alcohol-related liver disease. It is generally reversible if alcohol consumption is completely halted. However, its persistence may lead to more severe liver damage. According to a study involving hepatitis C patients, those who consume over 50 grams of alcohol daily show a 34% faster liver fibrosis progression than non-drinkers. Even moderate alcohol consumption, as low as 20 grams for women and 30 grams for men, may worsen liver damage in such patients. 2. Sugary snacks and beverages. Excessive consumption of sugary snacks may severely impact liver health, akin to the effects seen with excessive alcohol intake. Every cell in the body may metabolize glucose, but the liver is primarily responsible for fructose metabolism. When there's too much fructose, the liver converts much of it into fat through a process called lipogenesis. This may lead to fatty liver disease, a condition where excess fat is stored in liver cells. While a non-alcoholic fatty liver disease might initially be benign, it may progress to more serious liver damage if the dietary habits aren't corrected. Apart from that, a high intake of fructose may induce insulin resistance, a metabolic disorder where the body's cells fail to respond normally to insulin. Insulin resistance forces the pancreas to produce more insulin. It has several adverse effects, including prompting the liver to produce more fat. This exacerbates the accumulation of fat within the liver, potentially leading to a more severe condition called steatohepatitis. In fact, it also increases the risk of developing type 2 diabetes. According to research in children and adolescents, the relationship between fructose intake and non-alcoholic fatty liver disease, as well as the development of liver fibrosis, is dose-dependent, meaning the risk increases with the amount consumed. 3. Fried foods. Fried foods are typically high in trans fats, a type of unhealthy fat produced during the hydrogenation process to make cooking oils more stable. When these fats are consumed in large amounts, they may lead to an accumulation of fat in the liver. This condition, known as fatty liver, occurs when fat builds up within liver cells, impairing the liver's ability to function effectively. Over time, a fatty liver may become inflamed, leading to non-alcoholic steatohepatitis, a more severe form of liver disease that may progress to cirrhosis or liver cancer. Along with that, the high caloric content of fried foods contributes significantly to weight gain and obesity, both of which are risk factors for non-alcoholic fatty liver disease. Obesity promotes insulin resistance, which further exacerbates fat accumulation in the liver. This is because insulin resistance disrupts normal metabolic processes, causing the liver to store more fat rather than metabolize it. Additionally, the process of frying food often involves cooking at very high temperatures. leading to the formation of acrylamide and other harmful compounds. Acrylamide is a chemical compound that may form in some foods during high-temperature cooking processes, such as frying, roasting, and baking. These compounds may induce oxidative stress and inflammation in liver cells, compounding the effects of fat and exacerbating liver damage over time. In a study involving 74 overweight adolescents, 41% of adolescents with hepatic steatosis consumed more fried foods than those without hepatic steatosis. 4. White bread, rice, and pasta. Excessive consumption of white bread, rice, and pasta may be harmful to liver health due to their high glycemic index and refined carbohydrate content. The rapid conversion of these refined carbohydrates into glucose causes spikes in blood sugar levels. To manage this, the body releases insulin to help cells absorb the glucose and lower blood sugar levels. Over time, frequent insulin spikes may lead to insulin resistance, a condition where the body's cells don't respond effectively to insulin anymore. Insulin resistance is a key factor in the development of type 2 diabetes and is also associated with the accumulation of fat in the liver. This condition, known as non-alcoholic fatty liver disease, occurs when excess fats are stored in liver cells, significantly affecting the liver's ability to function normally. Moreover, the lack of fiber in these refined carbohydrates also plays a role in liver health. Fiber helps regulate blood sugar levels and supports healthy digestion. Without sufficient fiber, there is a higher risk of developing obesity and related metabolic diseases, including a further impact on liver health. A diet low in fiber may lead to increased intestinal absorption of fats, which are transported to the liver, increasing fat accumulation in the liver, and exacerbating conditions like non-alcoholic fatty liver disease. According to studies, Western dietary patterns, including white bread, are positively associated with non-alcoholic fatty liver disease. 5. Red meat. Research suggests that red meat and saturated fat may be associated with an increased risk of chronic liver disease and hepatocellular carcinoma, whereas white meat may be associated with a reduced risk. Red meat is rich in saturated fats, which, when consumed in large quantities, may lead to the accumulation of fat in liver cells. This condition, known as hepatic steatosis, is the initial stage of non-alcoholic fatty liver disease. Over time, if the diet remains unchanged and high in saturated fats from red meat, this may progress to more severe forms of liver disease. The liver, burdened with excessive fat, may become inflamed and start to scar, a condition known as fibrosis, which may eventually lead to cirrhosis, severely impairing liver function. Along with that, the high cholesterol content in red meat also plays a significant role in liver health. Cholesterol is essential for various bodily functions, but excessive amounts may contribute to fatty buildup within the liver. This not only exacerbates the effects of saturated fats, but also increases the risk of developing liver inflammation and subsequent liver damage. Managing cholesterol intake is crucial to preventing these harmful impacts on the liver. 6. Butter and margarine. Both butter and many types of margarine are high in saturated fats. When consumed in large amounts, saturated fats may lead to the accumulation of fat in liver cells, a condition known as fatty liver. This is the first stage of non-alcoholic fatty liver disease and may progress to more serious forms of liver disease if not managed. The liver's excessive fat buildup impairs its ability to function properly, reducing its efficiency in processing nutrients, hormones, and drugs and removing toxins from the body. Additionally, many margarines contain trans fats, which are created through an industrial process that adds hydrogen to vegetable oil, converting the liquid into a solid. Trans fats are especially bad for liver health because they cause inflammation and raise the risk of developing more serious liver diseases, such as steatohepatitis, inflamed fatty liver, and simple steatosis, fatty liver. Swapping out sources of saturated fat, like butter, for unsaturated fat sources, like olive oil, may be helpful for those with non-alcoholic fatty liver disease. This is one reason why the Mediterranean diet is sometimes recommended for individuals living with damaged livers. 7. Salt. Excessive salt in the diet leads to fluid retention because salt holds onto water in the body. This retention may cause swelling and increase the volume of blood, putting additional pressure on blood vessels and organs, including the liver. In individuals with liver disease, especially those with conditions like cirrhosis, fluid retention may be particularly problematic. It exacerbates symptoms such as ascites, where fluid accumulates in the abdomen, putting pressure on the liver and further impairing its ability to function efficiently. Moreover, high salt intake is linked to elevated blood pressure, known medically as hypertension. Hypertension is a risk factor for the development of fatty liver disease, independent of obesity. Increased blood pressure may damage the arteries in the liver, affecting liver blood flow. This damage may lead to more severe conditions, such as fibrosis, where normal liver tissue is replaced with scar tissue, ultimately diminishing the liver's ability to perform its vital functions. Along with that, salt may directly promote liver fibrosis, particularly in the presence of liver disease. High salt consumption has been shown to stimulate fibrosis in people with chronic liver disease by activating certain cells in the liver known as stellate cells. When activated, these cells produce excess collagen, leading to the buildup of scar tissue, fibrosis. As fibrosis worsens, it may progress to cirrhosis, severely impairing liver function and potentially leading to liver failure. In a study involving 310 type 2 diabetes patients, high salt intake, defined as over 9.5 grams per day, was linked to a higher prevalence of non-alcoholic fatty liver disease. Specifically, liver disease was found in 45% of patients with high salt intake, compared to 28% with lower intake. 8. Processed snacks. Many processed snacks contain trans fats, which are created through hydrogenation, a process that turns liquid vegetable oils into solid fats. Trans fats are harmful because they promote inflammation, a key player in liver damage. Chronic inflammation may lead to liver cell death and, over time, may cause fibrosis or scarring of the liver tissue. This not only impairs liver function but also sets the stage for more severe liver conditions. Additionally, processed snacks are often high in refined carbohydrates and sugars, which may lead to spikes in blood sugar and insulin levels. Overconsumption of these foods forces the liver to convert excess sugars into fat, contributing to fat accumulation in the liver, known as fatty liver. Additionally, persistently high insulin levels encourage the liver to produce and store more fat, exacerbating liver fat accumulation. Furthermore, the chemical additives and preservatives found in many processed snacks may also harm the liver. These substances, which include artificial flavors, colors, and preservatives, may be toxic to liver cells. The liver works hard to break down these chemicals to remove them from the body. However, excessive intake overloads the liver's detoxification capacity, leading to oxidative stress and damage to liver cells. This stress may impair the liver's ability to function properly and regenerate, further contributing to the risk of liver disease. A study involving 174,000 participants showed that higher ultra-processed food intake is associated with an increased risk of non-alcoholic fatty liver disease, liver fibrosis, and cirrhosis. 9. Artificial sweeteners. These sweeteners, found in many diet drinks, sugar-free snacks, and other low-calorie products, may contribute to liver damage, particularly when consumed in large amounts over long periods. Some artificial sweeteners may lead to disruptions in the body's natural metabolic processes. For example, there's growing evidence that certain sweeteners like aspartame and sucralose may alter the body's ability to process fats and carbohydrates. This disruption may lead to an increase in liver fat storage, a condition akin to what occurs with excessive sugar consumption. Artificial sweeteners may induce glucose intolerance and insulin resistance. Although they are not sugars, these sweeteners may still trigger insulin release, as the body anticipates sugar based on the sweet taste. Over time, this repeated insulin stimulation without actual sugar intake may confuse the body's metabolic controls, leading to insulin resistance. Insulin resistance is closely linked with increased fat accumulation in the liver and further exacerbates the risk of non-alcoholic fatty liver disease. Moreover, evidence suggests that artificial sweeteners may change the microbiota and cause dysbiosis, which may help non-alcoholic fatty liver disease develop. 10. High-fat dairy products. The high saturated fat content found in products like full-fat milk, cheese, and butter may lead to the accumulation of fat in liver cells, a condition known as hepatic steatosis or fatty liver. This occurs because the liver metabolizes dietary fats, and excessive intake overwhelms the liver's ability to process them efficiently. Over time, the ongoing accumulation of fat may cause liver inflammation, which may progress to more severe liver damage such as fibrosis, scarring of the liver tissue, cirrhosis, severe scarring and liver dysfunction, and even liver cancer if left unmanaged. Moreover, many whole-fat dairy products are also rich in cholesterol. An excess of dietary cholesterol may contribute further to the buildup of fat within the liver. High levels of cholesterol in the liver are associated with an increased risk of developing non-alcoholic steatohepatitis, a more aggressive form of fatty liver disease characterized by liver inflammation and damage. The results of a study involving about 7,500 participants clearly indicate that participants with higher fatty liver index scores consume more dairy, cheese, and yogurt, compared to the subjects with lower scores. Why should you be serious about liver damage? Liver damage is particularly harmful due to the essential role the liver plays in the body. As one of the body's largest and most complex organs, the liver has several critical functions that are vital for maintaining overall health. 1. Toxin removal. The liver is responsible for detoxifying harmful substances from the blood, including environmental toxins, food additives, alcohol, and medications. When the liver is damaged, it cannot effectively remove these toxins, which may then accumulate in the body, potentially leading to poisoning and other serious health issues. 2. Metabolism. The liver plays a central role in metabolizing carbohydrates, proteins, and fats. It helps regulate blood sugar levels by converting excess glucose into glycogen for storage and vice versa to maintain energy balance. Liver damage may disrupt these processes, leading to metabolic disorders such as hypoglycemia, low blood sugar, or hyperglycemia, high blood sugar, which are detrimental to overall health. 3. Digestion. The liver produces bile, a substance needed to digest and absorb fats and fat-soluble vitamins A, D, E, and K from the diet. A damaged liver may produce insufficient bile, leading to difficulties in digesting fats, which may cause nutritional deficiencies and problems with bowel movements. 4. Protein synthesis. The liver synthesizes various proteins, including albumin, which regulates blood volume and pressure, and clotting factors, which help blood to clot. Damage to the liver may lead to a decrease in the production of these proteins, resulting in symptoms like swelling in the legs due to low albumin levels and increased bleeding tendencies due to reduced clotting factors. 5. Immune system support. The liver is part of the immune system. It contains high levels of immune cells that act as a filter for the blood, capturing and breaking down bacteria, viruses, and other pathogens. Liver damage may weaken the immune barrier, increasing the risk of infection. While moderation is key in all aspects of diet, it's particularly important to manage intake of these foods to maintain liver health. Regular monitoring of your diet and lifestyle, coupled with periodic medical checkups, may help keep your liver functioning properly. Remember, a healthy liver means a healthier you. Now that we have discussed many foods that may damage our liver unknowingly, let's discuss other topics of interest, shall we? Watch 13 powerful benefits of eating oatmeal every day. You may also try our video on 10 types of foods that make your skin age faster. Go ahead, click one. Or, better yet, watch both. Will you willingly change your diet to protect your liver? Let us know in the comments below.